Hello students and welcome to the study portal. The topic of today's video is carbohydrate metabolism. So the carbohydrate metabolism is covered in BVMLT-204 subject, which is Introduction to Biochemistry, in its second unit. So we will be covering the second unit of BVMLT-204 today. Let's see the overview, the contents that of today's video that we are gonna cover in today's session. So first of all, we will having an introduction about the carbohydrate metabolism, that what is metabolism of carbohydrate and how it is carried out. Secondly, we will be talking about the classification of carbohydrates, that how the carbohydrates are classified and what all classes of carbohydrates are there. Then we'll be talking about the different pathways that are there through which the metabolism of carbohydrate is carried out, that is glycolysis, citric acid cycle, pentose phosphate pathway, gluconeogenesis, and the regulation of blood glucose. So we'll be covering about the different pathways in today's session. If there will be any need of second part, then we'll be covering the rest of the part in the second video as well. So let's start today's session without delaying anymore. So first of all, we'll talk about the nutrition. What is nutrition? So nutrition is defined as the science of how the body utilizes food to meet requirements for development, growth, repair, and maintenance. So nutrition is the science, is the technique, you can say, that how the body utilizes the food that is there in the body that we intake for its requirement to give the development, growth, repair, and maintenance. All right. So nutrients are the carbohydrates, fats, proteins, vitamins, minerals, and water that we intake. So the food we intake consists of different types of these kind of nutrients. So these nutrients are vitally required by our body for its growth, development, repair, and maintenance. So now talking about the carbohydrate metabolism, that what is carbohydrate metabolism? So it is a fundamental biochemical process which ensures a constant supply of energy to all the living cells. The most important carbohydrate is the glucose, which can be broken down via glycolysis, which enters then into the Krebs cycle and oxidative phosphorylation to generate the ATP, the energy which is required by our body to perform its different functions. So this glucose is a major carbohydrate which is a major fuel of tissues, which is converted into other carbohydrates through the different pathways that occur in the body. So carbohydrate metabolism is the whole of the biochemical processes which is responsible for the metabolic formation, breakdown, and interconversion of carbohydrates in the living organism. We can say that carbohydrates are central to many essential metabolic pathways. Plants, if we talk about plants, they synthesize carbohydrates from carbon dioxide and water through the process of photosynthesis. But if we talk about animal and fungi, they consume plants. They use cellular respiration to break down these stored carbohydrates to make energy available to all the cells. So humans can compose a variety of carbohydrates. Digestion breaks down complex carbohydrate into simpler ones, and after reabsorption in the gut, the monosaccharides are then transported through the portal vein to the liver, where all these non-glucose monosaccharides are transformed into glucose as well. Glucose is distributed to cells in the tissues where it is broken down by a cellular respiration or can be stored as glycogen. In cellular respiration, glucose and oxygen are metabolized to release energy with carbon dioxide and water as end products. Alright, so they are there. So carbohydrates, if we talk about the carbohydrates here, the carbohydrates are classified into 4 different classes on the basis of saccharide units, on the basis of polymerization. So what are carbohydrates? They are the polyhydroxy aldehydes or ketones. The aldehydes and ketone group is attached to them. So there are 4 different classes. First one is the monosaccharide, which consists of single sugar unit. Then is the disaccharide, which consists of 2 sugar units. Then the oligosaccharide, which consists of 2 to 10 sugar units. Then the polysaccharide, which consists of number of sugar units, many of the sugar units that can lead from 100 to thousands of the sugar units. So here you can see a more clear picture of the classification of carbohydrate is given here. Monosaccharides are there, disaccharides are there, oligosaccharides are there, and saccharides are there, and each example is given against them. So glucose is an example of a monosaccharide, sucrose, maltose, lactose are an example of the disaccharides, raffinose, thiokose are the example of oligosaccharides, dextrin, starch, cellulose, pectin, gums are the example of polysaccharides. Now talking about the monosaccharides. So depending upon the carbon or aldehyde or atom's ketone group, monosaccharides are there. So monosaccharide can consist of 2 to 6 carbon units. You see, so they have a single sugar unit name based on the carbon unit and aldehyde or ketone group. They are classified as triose, aldoses, tetrose, ketose, in case of ketosis, pentoses, and hexoses. Then are the disaccharides. They consist of 2 sugar units. They can be maltose or the maltotriose or the saccharose. Now talking about the metabolism. What is metabolism? So the entire spectrum of chemical reaction which occurs in the living system is referred to as the metabolism. We can say that metabolism is the process of metabolism is the process of breaking down of it is a series of chemical reaction which occurs inside the cell. The reactants, products, and the intermediates of an enzymatic reaction, metabolites, which are modified by a sequence of chemical reactions which is catalyzed by the enzymes, we can say. So the metabolic reactions, there are 2 types of metabolic pathways that are characterized by their ability to either synthesize molecules with the utilization of energy or break down complex molecule and release energy in the process. So there are anabolic pathway, catabolic pathways, and the amphibolic pathways. Now what are anabolic pathways? So in the processes where the molecule is synthesized with utilization of energy, it is called as the anabolic pathways. But where the molecule complex molecule is broken down into simpler process and the energy is released is called as the catabolic pathway. Now amphibolic pathway, in amphibolic pathway some of the energy is released and some of the energy is gained. So it is a mixture of both anabolic and catabolic pathway. So it is called as the amphibolic pathway. Now here you can see the main pathways of carbohydrate metabolism. So there is you can say glycolysis is there, gluconeogenesis is there, glycogenesis, pentose phosphate pathway, glycogenolysis, in which the lactate is formed, or you can say the pyruvate is formed. Here you can see the complete pathway of the carbohydrate metabolism. We will be talking about all of these pathways in detail in today's video. So the food molecules are the simpler molecules, then amphibolic pathways there, anabolic catabolic pathways there, which may result in the production of carbon dioxide and water and the carbohydrates. So metabolic pathways may be studied at different level of organization. The organizational level can be tissue level and the subcellular level as well. So what are carbohydrates? So carbohydrates, they are the primary source of energy in the cell. They are central to all metabolic processes. What happens? Glucose enters into the cytosol, anaerobic pathway, hexokinase is there. Hexokinase occurs and the hexokinase enters the pentose phosphate pathway and it will give the glucose 6 phosphate. Then it will enter the phosphate shunt, then glycose occurs. Then the glycogen is formed. Then the glycogen breaks down into pyruvate through which energy is released into the body. Now talking about glycolysis, the first pathway. So glycolysis is the process of breaking down a glucose molecule into two pyruvate molecules while storing energy released during this process as ATP and NADH. So we can describe it as a sequence of reaction of glucose to lactate and pyruvate with the production of ATP. Alright. So if you will break the word glycolysis, you will get glyco, which means sweet or sugar, and lysis, which means dissolution or breaking down. So breaking down of glucose is called as glycolysis. Now, what is the site of glycolysis? So glycolysis occurs in the cytosolic fraction of the cell. It starts at the cytosolic fraction of the cell, but somehow it also enters into the mitochondrial stage and then it occurs. So we can say nearly all organisms that break down glucose utilize glycolysis. Glucose regulation and products used are the primary category in which these pathways differ between organisms. In some tissues and organisms, glycolysis is the sole method of energy production. This pathway is common to both aerobic and anaerobic respiration. So as you can see in the picture, glycolysis consists of 10 steps, which is split between two phases. During the first phase, you can see it took the breakdown of two ATP molecules. During the second phase, chemical energy from the intermediates is transferred into ATP and NADH. The breakdown of one molecule of glucose results into two molecules of you can see pyruvate, which can be further oxidized to access more energy in later processes. So glycolysis can be regulated at different steps of the process through the feedback regulation. The step that is regulated the most is the third step here, you can say, because it is the irreversible step. Alright, so this regulation is to ensure that the body is not overproducing the pyruvate molecules. The regulation also allows for the storage of glucose molecules into fatty acids. So there are various enzymes that are used through glycolysis. The enzyme upregulate, downregulate, and feedback regulate the process, we can say. Alright, so this is the process of this is the metabolic pathway which converts the glucose into pyruvic acid. The free energy released in the process is used to form the high energy molecules which is ATP and NADH. It is a sequence of 10 reactions which is catalyzed by the enzyme. So glycolysis is a metabolic pathway that does not require oxygen. The wide occurrence of glycolysis in other species indicate that it is an ancient metabolic pathway. Alright, so it occurs in most organisms. It occurs mainly in the part of the cells, the cytosol. Alright. So let's have an overview at the sequence of reaction. So first of all, glucose is there. With the help of hexokinase, glucose is converted into glucose 6-phosphate. With the help, then the PGK will act and it will convert it into glucose 6-phosphate. From here, this glucose 6-phosphate is then utilized with the help of glucose 6-phosphate dehydrogenase enzyme, and it will enter into the pentose phosphate pathway where it will be converted into the xylulose 5-phosphate. Alright, here with the help of PGI, glucose 6-phosphate will convert into the fructose 6-phosphate. With the help of PFK for phosphofructokinase, this fructose 6-phosphate will convert into fructose 1,6-biphosphate. Then with the help of aldolase, the two molecules will be broken down. First will be glyceraldehyde 3-phosphate and the second one will be DHAP, which is dihydroxyacetone phosphate, thereby releasing a molecule which is NADH. Alright, so here then it will convert with the help of enzyme into 1,3-biphosphoglycerate. Then with the help of PGK is converted into ATP. One molecule of energy is we get from there. Thereafter 3-phosphoglycerate will convert into 2-phosphoglycerate. Then with the help of enolase and magnesium ions, phosphoglycerate will get converted into phosphoenol pyruvate, and there ADP will get converted into ATP. Then 1 molecule of energy is gained from here. So in total we get 3 molecule of energy. 2 are the ATPs and 1 is the NADH. So if there is presence of oxygen, this phosphoenol pyruvate will get converted into pyruvate. But there is absence of oxygen, this pyruvate will get converted into the lactate. So this is how the glycolysis is carried out. So the first 5 steps of glycolysis are regarded as the preparatory phase, since they consume energy to convert the glucose into 3 carbon sugar phosphates. Alright, but the later steps, in the later steps we get the production of energy molecules. 3 energy molecules are produced, due to which we get the energy from the glycolysis cycle. Alright, so here you can see total of 8 ATPs are formed in the glycolysis process. Oxidation of glucose in aerobic condition may generate 38 ATPs and in anaerobic condition 2 ATPs. So what is the biomedical importance of glycolysis now? So glycolysis is the principal route of metabolism. It is a principal pathway through which energy is produced and we get ATPs. So it is also used for the production of acetyl CoA in the citric acid cycle, because as you can see, the glucose 6-phosphate is taken from here, and the other biomolecules are also taken from the glycolysis pathway only. So it is also the major cycle for the metabolism of fructose and galactose. It also provides ATPs in the absence of oxygen. Oxygen is not mandatory for this process, so thus we get the production of antibodies, thus we get the production of ATPs, energy molecule, even in the absence of oxygen. If we see the clinical aspects of the glycolysis, we can see that in case of hemolytic anemias, inherited aldolase A and pyruvate K deficiencies can be seen. Skeletal muscle fatigue is there. Inherited pyruvate dehydrogenase deficiency may lead to the lactic acidosis also. And fast growing cancer cells, glycolysis proceeds at the faster rate, which may result in the increased acidic environment in the body and may lead to the implication of certain types of cancer. So this is the biomedical importance of the glycolysis process. So the final common pathway for oxidation of carbohydrates, lipids and protein. It also has a major role in gluconeogenesis and production of glucose, transamination, deamination and lipogenesis. The vitamins also play a key role in this cycle. Example, riboflavin and FAD, niacin in production of NAD and thiamine. Alright, so thus glycolysis is very important process in the whole carbohydrate metabolism, because it will give different type of molecules of energy. It takes place in the cytoplasm of a cell and does not require oxygens. Alright, so why it is important? It is important in the cell because glucose is the main source of fuel for the tissues in the body, and if you see, the glucose is broken down and the energy is carried out by the glucose by the process of glycolysis, you can say. So it is the essential metabolic pathway. Major themes which you have to remember is it is a sequence of enzymatic reaction. ATP is invested in this and then recapture net gain of 2 ATPs. So it is the first step of cellular reaction where glucose gets converted into pyruvate or lactate. Alright. So if we talk in cellular respiration, how glucose is broken down and gives the production of energy is what we can see with the help of glycolysis. So it is to ensure normal brain function, the body must maintain a constant supply of glucose in the blood. So it is also important because the metabolism of glucose produces useful intermediates for other metabolic pathways such as the synthesis of amino acids or fatty acids. Thus glycolysis is of major importance. We will study about the different cycles, the other cycles of the carbohydrate metabolism in another session. Thank you for joining.
Hello student, in this video we discuss about the topic glycolysis and Krebs cycle. That is very important topic in the biochemistry metabolism. Okay. First of all, you see this map. What you see this map? What this map tell about? Actually, this map contain all the metabolic pathway that was occur in our body in biochemistry. That all metabolic pathway are present in this diagram that called metabolic metro map. All metabolic pathway are connected with each other, and that all are present here. See, this is carbon fixation, this is pentose phosphate pathway, fatty acid synthesis, fatty acid elongation, beta oxidation, citric acid cycle, this is glycolysis, this process is the glycolysis, and this opposite of glycolysis is gluconeogenesis. And this glycolysis pyruvate, that is the product of glycolysis, is connected with the citric acid cycle with the help of acetyl coenzyme A. And this, with the help of this citric acid cycle, the urea cycle is connected, right? And with this urea cycle, amino acid deamination will occur. See here, here is amino acid deamination process. Okay. By this citric acid cycle, gluconeogenesis is connected with this glyoxylate cycle. So all metabolic pathway are connected with each other. The product or subproduct of the process. That subproduct are connected with each another type of cycle, like fatty acid synthesis. See here, the citrate of the citric acid cycle is connected with the fatty acid synthesis, right? And here this is connected to beta oxidation, acetyl coenzyme A. Okay. And again the product of the subproduct of beta oxidation is connected with waxes, sphingolipid. So all the metabolic pathway that occur in our body or in other organisms, all are connected with each other, and that all pathway are start with which substrate. All metabolic pathway are started with which substrate? That is glucose. What? That is glucose, fructose, like the glucose or fructose. All metabolic process are started with the glucose or fructose. That is glycolysis. So starting of the all metabolic pathway is the glycolysis. That occur where? Glycolysis occurred where? Cytoplasm. Right? So glycolysis is the cytoplasmic pathway because they occur in a cytoplasm. That's why it is called as a cytoplasmic pathway. What? Cytoplasmic pathway. Glycolysis is the cytoplasmic pathway which break down glucose into 3 carbon compound and generate energy. What do the glycolysis? In glycolysis, 6 carbon compound, that is glucose, is break down into 3 carbon compound, that is pyruvate, right? And this process is known as glycolysis. This process is known as glycolysis. So, come, what is a glycolysis? Actually, glycolysis is the first stage in a cellular respiration. Already I told, glycolysis is the first step, means starting of the all metabolic pathway. Starting of the all metabolic pathway is the glycolysis. This is the cytoplasm, and here the process of glycolysis will be takes place. Understand? So, Greek glycolysis. Glycolysis is made up of what? Glycose and lysis. Glycose and lysis. Glycose means sugar, sweet in nature, right? And lysis means breakdown or dissolution. So, what do you mean by glycolysis? Means breakdown of sugar. What? Glycose means sweet, sugar, and lysis means breakdown. Then what happened? Breakdown of the sugar. Breakdown of sugar is known as glycolysis. So, glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. For cellular metabolism, that energy are in the form of ATP. What? ATP and NADP. Nucleotide adenine diphosphate. Right? This is the ATP and this is the NADP. That is the form of energy. Glycolysis consists of energy requiring phase followed by energy releasing phase. Actually, in glycolysis, there are two phases will be occur. In first phase, energy are used. Means ATP are used in first step. And in second step, that is called, first step is known as energy requiring phase. Energy requiring phase, because they utilize the ATP. They use the ATP. That's why that phase is known as energy requiring phase. And next phase is the energy releasing phase. What? Energy releasing phase. In this phase, ATP is released and NADH is released. Okay? ATP and NADH is released here. So, this stage is known as energy releasing stage. So, overall production of energy in glycolysis is 2 ATP and NADH. With the help of one NADH, with the help of oxidative phosphorylation, this NADH is converted into ATP. By which process? Oxidative phosphorylation. One NADH, with the help of one NADH, 2.5 ATP will be formed. What? 2.5 ATP. So, in glycolysis, the overall production of ATP is 2 ATP and 2 NADH. Then you convert that and get the total. Okay? This is the process. Glucose. This is a glucose 6-carbon molecule in cytoplasm. They converted into pyruvic acid plus energy. That is 3-carbon molecule. This is pyruvic acid. That is 3-carbon molecule. Another process. This process is known as glycolysis. Right? This process is known as glycolysis. In next process, this glucose molecule is converted into ethanol. That is 2-carbon. That is 2-carbon molecule plus CO2 and plus energy. This process not require oxygen. in the absence of oxygen, and in this process they require oxygen. So this is known as fermentation. What? Fermentation. This process is known as fermentation that occur in a yeast. Another is a lack of oxygen molecule in cell. They converted into a lactic acid, that is 3 carbon plus energy. This process is occur in a muscles. Right? So these are the three processes by which glucose molecule can be break down. First process is the glycolysis. First process is glycolysis. First process is glycolysis that occur in presence of oxygen. Second is fermentation. Fermentation that occur in the absence of oxygen and converted into ethanol. And third is lactic acid formation. Lactic acid formation that also occur in the absence of oxygen and form product that is known as lactic acid. So these are the 3 steps by which glucose molecule can be break down. Here we study of the process of glycolysis. Okay, here we study the glycolysis. Actually, in all 3 processes, energy will be released, either in the form of 1 ATP or 2 ATP or 3 ATP, but energy will be released in all process. Here, glycolysis is the series of reaction that extract energy from glucose by splitting it into 3 carbon molecule that is called as pyruvate. Sequence of reaction converting glycogen to pyruvate, lactate or ethanol with the production of ATP as energy. ATP as the energy form. Reversal of glycolysis along with the alternate arrangement of the irreversible steps will result in the synthesis of glucose, that is known as gluconeogenesis. Gluconeogenesis is nothing but it is a reversible process of glycolysis. Reversible process of glycolysis is known as gluconeogenesis. Reversible process of glycolysis known as gluconeogenesis, that is from pyruvate to glucose. Pyruvate to glucose is known as gluconeogenesis. Okay, clear here? This is the steps of glycolysis and energy regeneration. This is glucose molecule. After breaking of glucose molecule, 2 pyruvate molecule will be generated. Remember this, because this is a 6 carbon molecule and pyruvate is the 3 carbon molecule. That's why total 6 carbon molecule, then 2 pyruvate will be generated. This pyruvate molecule is converted into acetaldehyde, after that ethanol. What? After that ethanol. Then this process is known as fermentation. This step is known as fermentation. In organism that perform cellular respiration, glycolysis is the first stage of this process. In every organism, breakdown of glucose is the first step of the metabolism. Because we eat anything that will be in the form of glucose. We take everything in the form of glucose. So every reaction will takes place in our body or in other organism body after the breakdown of glucose, either in the presence of oxygen or either in the absence of oxygen. But there is a breakdown of glucose will be happen. However, glycolysis doesn't require oxygen, and many anaerobic organisms that do not use oxygen also have this pathway. Glycolysis takes place in the cytosol of a cell, and it can be broken down into three main phases. Glycolysis takes place in the cytosol, right? And it can be broken down into three main phases. That is, first energy requiring phase, second is splitting phase, third is energy releasing phase. First is energy requiring phase, second is splitting phase, where actually splitting phase is not main, it is not important, but energy requiring phase and energy releasing phase is important. Splitting phase is the one step where glyceraldehyde 3-phosphate, 2 molecule of glyceraldehyde 3-phosphate will be formed. That is one step in the glycolysis. That's why it is not important. You remember only energy releasing phase and energy requiring phase. Glycolysis is the first step in a cellular respiration. Remember this. Here is a pathway of glycolysis. Glucose molecule is break down into 6. Actually, one thing that several enzymes are used. What? Several types of enzyme. Several type of enzyme are used here. Like hexokinase, phosphoglucosisomerase, phosphofructokinase, aldolase, glyceraldehyde 3-phosphate, dehydrogenase. Remember this. If NADH, formation of NADH will be takes place, then dehydrogenase enzyme are used. For formation of NADH, dehydrogenase enzyme are used, and the formation of ATP kinase are used. Remember this thing. For NADH, dehydrogenase enzyme are used, and for ATP, kinase enzyme are used. These are important. So, first step, glucose. Glucose is converted into glucose 6-phosphate. During this conversion, glucose to glucose 6-phosphate, 1 molecule of ATP is required. What? 1 molecule of ATP is required here. This is a glucose molecule. This glucose molecule is converted into glucose 6-phosphate. 1, 2, 3, 4, 5, 6. So here 1 phosphate group is attached. During the attachment of 1 phosphate group, 1 ATP is required here. What? 1 ATP is required here. Understand? Next step is conversion of glucose 6-phosphate to fructose 6-phosphate. Glucose 6-phosphate to fructose 6-phosphate. So here what happened? Here is isomerization. Nothing will change. Only isomer will be changed. Right? Glucose 6-phosphate to fructose 6-phosphate. Here the 6 molecule cycle is present. Here 5 carbon cycle. Here 6 carbon cycle. Here 5 carbon cycle. So here isomerase enzyme is used, that is phosphoglucoisomerase. And here there is no requirement of ATP, and there is not a release of ATP. Okay. Next step is the conversion of fructose 6-phosphate to fructose 1,6-biphosphate. Means there is addition of one phosphate molecule on another carbon. Means during the addition of one phosphate, remember during the addition of one phosphate molecule, one ATP is required. One ATP is required. In the first step, at the 6th position phosphate group is added, then one ATP is required. Now at first position, one phosphate molecule is added, means one ATP is required here. Right? So here fructose 1,6-biphosphate will be formed. After that, this step is known as energy. This step is required energy, so this is known as energy requiring step. Energy requiring step. Because in this step, at two time, first and third, 2 ATP is required. Now this is known as, after fructose 1,6-phosphate, this is known as splitting phase. Here fructose 1,6-phosphate is splitted into 3 carbon molecule, that is glyceraldehyde 3-phosphate. That is glyceraldehyde 3-phosphate. See, 1, 2, 3. This is 3 carbon molecule. So this step is a splitting step. After this glyceraldehyde 3-phosphate, another molecule is formed, that is 1,3-bisphosphoglycerate. And remember, in the splitting phase, there are 2 molecules. Remember this, 2 molecule of glyceraldehyde 3-phosphate is formed. 2 molecule of glyceraldehyde 3-phosphate is formed. So after the 5th step, all the process will take at the 2 times, because there 2 molecule, 3 carbon 2 molecules are present, right? So each and every step will take at the 2 times. After the formation of glyceraldehyde 3-phosphate, this glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. 2 molecule of 1,3-bisphosphoglycerate. During the formation of this, one molecule of NADH is released. Remember this. One molecule of NADH is released. Already told, during the formation of NADH, which enzyme is used? Dehydrogenase. During the formation of NADH, dehydrogenase enzyme is used. And there is 2 molecule of glyceraldehyde, means 2 in 1 NADH molecule is multiplied by 2, means 2 NADH is used, is formed. Not used, it is 2 molecule of NADH is formed. Right? After 1,3-bisphosphoglycerate, this 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Means there is a conversion or transferring of phosphate group will be takes place here, right? So during the transfer of the phosphate group, one ATP is formed. During the addition of phosphate group, ATP is used, right? and during the removing of the phosphate group, one ATP is formed. Remember this. During addition of the phosphate group, energy will be required, and during the removal of the phosphate group, energy will be released. Remember this is. So 1,3-bisphosphoglycerate will convert it into 3-phosphoglycerate. This formation will take like this enzyme kinase, because here is a removal of one phosphate molecule, and during the removal of the one phosphate molecule, one molecule of ATP is formed here. And we remember that here is 2 molecule of 1,3-bisphosphoglycerate is present. So 2 ATP is formed here. After the formation of 2-phosphoglycerate, it is converted into phosphoglycerate, 2-phosphoglycerate. Here only mutarotation will take place, and it is the enzyme that is used is mutase. Another is a anolase enzyme is used, and phosphoenol pyruvate will be formed. During the formation of phosphoenol pyruvate, they also contain phosphate group. So we want to remove all phosphate group. So during the removal of the phosphate group and formation of pyruvate, one ATP molecule will be released. And the enzyme that is used here is a pyruvate kinase. Okay, understand? Clear this? This is all 10 steps. In glycolysis, total 10 steps will be there. Okay. How much energy will be formed here? See here. ATP used, that is 2 ATP, right? 2 molecule of ATP will be used, that is -2. ATP that is formed is here 2 ATP will be formed, and here 2 ATP will be formed. Total 4 ATP plus 4 ATP. And NADH, 1 molecule of NADH will be formed in each step, means 2 NADH molecule will be formed. Two molecule of NADH will be formed, and with the help of one NADH, how much ATP will be formed? 2.5, means with the help of 2 NADH, 5 ATP will form. Then how much total ATP energy will be released in glycolysis? 5 plus 4, 9, 9 minus 2, 7. 7 molecule of ATP overall. 7 molecule of ATP will be formed in glycolysis, right? Understand? It is clear? Clear this is? So next is here energy requiring phase. Already understood what happened in energy requiring phase? This is 1 molecule of glucose, and in energy requiring phase, this 1 molecule of glucose is converted into 2-glyceraldehyde 3-phosphate, and during the formation of this 2-glyceraldehyde 3-phosphate, 2 ATP will be required, right? During the addition of phosphate group at the 1 and 6 position of the carbon, 2 ATP will be required. So this process is known as energy requiring reaction. What? This process is known as energy requiring reaction. In this phase, the starting molecule of glucose gets rearranged and 2 phosphate group, remember 2 phosphate group are attached to it. The phosphate group make the modified sugar, now called as fructose 1,6-bisphosphate, that is unstable, allowing it to split it in half and form 2 phosphate bearing 3 carbon sugar. because the phosphate used in this step come from ATP. Two ATP molecule get used here, right? Already I told you that during the addition of phosphate group at one carbon position and 6 carbon position, 2 ATP molecule are used, right? Clear? So this process is known as energy requiring phase. Next step is splitting step. Splitting step means where fructose 1,6-bisphosphate or diphosphate is dissociated into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and this dihydroxyacetone phosphate is again converted into glyceraldehyde 3-phosphate. This process is known as splitting step. Another is an energy releasing phase. In energy releasing phase, glycerol with the help of glyceraldehyde 3-phosphate converted into 2 molecule of glyceraldehyde 3-phosphate converted into 2 pyruvate, that is 3 carbon compound. During the conversion of the glyceraldehyde 3-phosphate to 2 pyruvate, 2 molecule of NADH and 4 molecule of ATP will be formed, because phosphate molecule are removing. 2 phosphate molecule are added in earlier, 2 phosphate molecule are added, right? So now we want to remove that phosphate molecule for stability. When you removing this phosphate group, ATP will be released. So there is two molecule of glyceraldehyde 3-phosphate will be formed. Means with the help of 1 molecule of glyceraldehyde, 2 ATP will be formed. So 2 molecule means 4 ATP will be formed, right? So this process is known as energy releasing reaction. In this phase, each 3 carbon sugar is converted into another 3 carbon molecule, that is pyruvate, through a series of reaction. In this reaction, 2 ATP molecule and 1 NADH molecule are made, because this phase takes place twice, because 2 molecule of glyceraldehyde 3-phosphate will be formed. Once for each of the 3 carbon sugar, it makes 4 ATP and 2 NADH overall. Understand? This step is clear? This is the overall glycolysis that convert 1,6 carbon molecule of glucose into 2,3 carbon molecule of pyruvate. The net products of this process are 2 molecule of ATP and 2 molecule of NADH, right? See, this is actually in metabolic reaction there are 3 stages. That is, first stage is glycolysis. Second step is conversion of pyruvate to acetyl coenzyme, and third step is TCA cycle, that is tricarboxylic acid cycle. So in all steps the energy will be released. Overall energy will be released in this all step. So now we study about the first step. Here we study about first step, that is glycolysis. In glycolysis, overall energy is 2 ATP and 3 ATP, net yield 2, because 2 ATP is already utilized in first stage, that is energy requiring stage, right? So here is 2 ATP, and with the help of NADH 3 ATP will be formed, but actual from 1 NADH 2.5 ATP will be formed. Means with the help of 2 NADH 5 ATP will be formed. Okay, clear? How many ATP? 5 ATP. So overall energy in glycolysis is 7 ATP. this is the step. This is the step. That is, glucose is converted into glucose 6-phosphate with the help of, these are the steps that is in detail. These are the glucose. Glucose is converted into glucose 6-phosphate with the help of hexokinase. Glucose 6-phosphate is converted into fructose 6-phosphate with the help of phosphoglucose kinase, and fructose 6-phosphate is converted into fructose 1,6-bisphosphate, means addition of one phosphate molecule with the help of kinase molecule. And this step is the splitting step. What? This is the splitting step. Splitting step means the fructose 1,6-phosphate molecule is split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and again dihydroxy phosphate is converted into glyceraldehyde 3-phosphate. Okay. In where ATP is added means step where phosphate is added means ATP is utilized. Remember this. And kinase enzyme are used. Okay. Understand? This is the energy requiring phase. This are a step where which enzyme is used, which are isomer, like that only. Already I told you this all steps. First step where glucose is converted to glucose 6-phosphate, one phosphate molecule is added, one ATP is utilized here. In second step, glucose 6-phosphate is converted into the isomer, that is fructose 6-phosphate. In third step, another phosphate group is added at one position of the carbon of the fructose, right? And one ATP is utilized here, right? In fourth step, fructose 1,6-bisphosphate is split into two molecules, that is DHAP and glyceraldehyde 3-phosphate. And in fifth step, DHAP is converted into glyceraldehyde 3-phosphate because it is a stable product. DHAP is not stable. That's why it's converted into glyceraldehyde 3-phosphate. Another this step is a energy releasing step. In this step, glyceraldehyde 2 molecule of glyceraldehyde 3-phosphate that is formed is converted into 1,3-bisphosphoglycerate, and here dehydrogenase enzyme is used and NADH will be formed. Means one molecule of NADH will be released here. 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Here one phosphate molecule will be removed. One phosphate molecule will be removed here. This phosphate molecule or this phosphate molecule will be removed here. Means one ATP will be released, and in this process one ATP one phosphate molecule will be removed. In this is removed. Here also one ATP will be formed and converted into pyruvate molecule. So these are steps are the energy releasing step. Already 6, 7, 8, 9 steps are you already know. Two half reaction occur simultaneously, means glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Then after that 1,3-phosphoglycerate is converted into 3-phosphoglycerate, right? One ATP will be formed here. In above step, one NADH is formed and dehydrogenase enzyme are used there. In 8th step, mutation will be takes place or isomer will be formed, that is 2-phosphoglycerate. In 9th step, 2-phosphoglycerate loses one molecule of water molecule and form a phosphoenol pyruvate, that is PEP. In last step, removal of the phosphate group will be takes place, removal of phosphate group and releasing of one ATP molecule, and that converted into a stable product, that is a pyruvate. Pyruvate is the end of the glycolysis. Actually, this you recall all the step of the glycolysis. During the recalling, there are inhibitor are also present that inhibit the process of the glycolysis. Not at step of the glycolysis inhibitor is the present. There are several steps where inhibitor are go and bind with that enzyme that convert the substrate into product and inhibit the all process, like arsenate, iodoacetate, arsenate and iodoacetate. They inhibit the arsenate and iodoacetate inhibit the glyceraldehyde 3-phosphate dehydrogenase enzyme. Inhibit the glyceraldehyde 3-phosphate dehydrogenase enzyme. Another is bromohydroxyacetone phosphate. They inhibit the dihydroxyacetone phosphate that is formed during at the splitting stage, right? During the inhibition of formation, they inhibit the dihydroxyacetone phosphate, means one molecule of pyruvate will be formed only, because during this splitting of the fructose 1,6 bisphosphate, two molecules will form. One is glyceraldehyde 3-phosphate and another is dihydroxyacetone. And after that, this dihydroxyacetone will be converted into glyceraldehyde 3-phosphate. When bromohydroxyacetone inhibit this dihydroxyacetone phosphate, they not converted into glyceraldehyde 6-phosphate, means one molecule one step will be inhibited, means one molecule of pyruvate will be formed only, not two molecule of pyruvate. Another is fluoride. Fluoride inhibit enolase. Enolase are used where removal of one molecule of water will be takes place, means at the 9th position. When they inhibit the PEP, formation of PEP enzyme, if PEP molecule will be not formed, then pyruvate will be not formed, means the inhibition of total 2 pyruvate molecule, means 2 pyruvate molecule will be not formed, because fluoride inhibit the enolase. That is 9th step of the glycolysis, 9th step. Another is oxamate. They inhibit the lactate dehydrogenase. Actually, lactate dehydrogenase are not used in this glycolysis. They used during the formation of lactic acid. During the formation of lactic acid, not in this 10th step. In 10th step, iodoacetate, bromohydroxyacetone and fluoride. These are the main inhibitor in the glycolysis. Right? In glycolysis, arsenate iodoacetate inhibit the glyceraldehyde 3-phosphate. If glyceraldehyde 3-phosphate dehydrogenase will be not formed, they inhibit the formation of glyceraldehyde 3-phosphate dehydrogenase enzyme, means there is no formation of the pyruvate molecule will be takes place. Or another step after that, this is the 6th step, right? 6th step of the glycolysis. After 6th step, rest 4 step will be not takes place, right? This is the step where inhibitor are present and inhibit the process of glycolysis. Here is this red see, this is a pyruvate kinase. This is a phosphofructokinase. Okay. So here glucose 6-phosphate inhibit the hexokinase 1. This is the overall inhibitor after TCA cycle also, the overall inhibitor that inhibit the glycolysis process. The ATP citrate that form that will inhibit phosphofructokinase, like that. But we study that in earlier slide, in previous slide, we studied that enolase. This enolase is inhibited by fluoride. Okay, means this step will be not takes place after inhibition, right? Iodoacetate inhibit the dehydrogenase enzyme, right? At this step, this is inhibited by iodoacetate, right? Means this process will be stopped. Bromide will be inhibit the DHAP. So these are the inhibitory product. This all about the process of glycolysis. Now come with at the TCA cycle. What is TCA cycle? TCA is the tricarboxylic acid. What? Tricarboxylic acid cycle. TCA is located in a mitochondrial matrix in eukaryotes. In prokaryotes, it's located already in a cytoplasm. What is the main goal of the TCA cycle? The main goal of TCA cycle is the breakdown of pyruvate into a CO2 and acetyl coenzyme A and release more energy. What happened here? Break down the pyruvate or pyruvic acid into a CO2 plus energy, so that more energy will be formed. Next, what is the process? Each pyruvate loses one carbon and make a 2 carbon molecule, that is acetyl coenzyme. Actually, before TCA cycle, one step is there where pyruvate is converted into a acetyl coenzyme A, because pyruvate is a 3 carbon molecule. They in TCA cycle require 2 carbon molecules, so they converted into a 2 carbon molecule, that is acetyl coenzyme A. The carbon join with the oxygen that breathe in to create the carbon dioxide we exhale. The acetyl coenzyme can then diffuse into the matrix of the mitochondria. This acetyl coenzyme may go into a mitochondrial matrix, and where TCA cycle will be takes place, right? This is the TCA cycle. Here what happened? This is a pyruvate, that is the product of the glycolysis. This pyruvate is a 3 carbon molecule converted into a 2 carbon molecule, that is acetyl coenzyme A. This acetyl coenzyme A go and in TCA cycle in the mitochondrial matrix and combine with citrate, combine with oxaloacetate and form a citrate. This citrate is 1, 2, 3, 4, 5, 6 carbon molecule. Then is converted into cis-aconitase. Cis-aconitase converted into diisocitrate, then alpha-ketoglutarate, then succinyl coenzyme A, then succinate, fumarate, malate, oxaloacetate. This all step will be continuously takes place after the addition of acetyl coenzyme A. So during the formation of this process, during the process of this cycle, citric acid cycle, here NADH, FADH and GTP or ATP will be formed here. So more energy will be released here. What? More energy will be released here. Here, see, during the conversion of diisocitrate to alpha-ketoglutarate, one molecule of NADH will be released. During the conversion of succinyl coenzyme A to succinate, one ATP or GTP will be formed. One GTP will be formed here, right? And during the formation of fumarate to malate, one FADH will be released. And during the malate to oxaloacetate, one NADH will be released. So overall production of the NADH FADH is the 3 NADH, 1 ATP and 1 FADH will be formed here. So huge amount of energy will be released here. This NADH, the NADH that are formed in glycolysis or TCA cycle, this all NADH and FADH go into a oxidative phosphorylation. Go into oxidative phosphorylation for the formation of ATP. For the formation of ATP. Right? This FADH, NADH go into a process where oxidative phosphorylation, that process where ATP will be formed from this NADH and FADH. Right? These are the process of the TCA cycle. Now, since there are two molecule of acid, remember that two molecule of pyruvate will be formed. Means two molecule of pyruvate will be formed. Then two molecule of pyruvate will be converted into two molecule of acetyl coenzyme A, and two molecule of acetyl coenzyme A will enter into TCA cycle. Means TCA cycle will takes place at a Krebs cycle, or TCA cycle takes place at 2 times. What? 2 times. So, there must be 2 turn of the cycle will takes place, one for each pyruvate. This is the one cycle by one pyruvate. This is the another cycle by another pyruvate. Okay, clear? Next, what is the reactant and what is the product? Product from glycolysis. What is the product of the glycolysis? 4 carbon molecule will be recycled. What is the product of acetyl coenzyme A? ATP. Sorry. Here, what is the product of glycolysis? The product of glycolysis is the pyruvate. Okay. Product of acetyl coenzyme is the 4 carbon molecule that is recycled. Product of ADP is ATP, and product of oxygen is carbon dioxide. FAD is FADH2, and NAD plus is NADH. Okay. So these all are the product. What is the reactant? What is the product? What happened? Next is inhibitors of inhibitor in citric acid cycle. The inhibitor of citric acid cycle is fumarase. What? Inhibitor of citric acid cycle is malonate, arsenate, and fluoroacetate. Fluoroacetate inhibit the aconitase. Arsenate inhibit the alpha ketoglutarate dehydrogenase, and malonate inhibit the succinate dehydrogenase. Fumarase is an enzyme, fumarase is not inhibitor. Fumarase convert the fumarate into an L-malate. So there are three inhibitor in the citric acid cycle. First is fluoroacetate, second is arsenate, and third is malonate. Okay. Next is significance of TCA cycle. TCA cycle are a complex oxidation of acetyl coenzyme. They are help in a generation of huge amount of energy, that is form ATP, NADH or FADH. Final common oxidative pathway, integration of major metabolic pathway. Fat is burned on the way of carbohydrate. Excess carbohydrate are converted as neutral fat, and no net synthesis of carbohydrate from fat. Carbon skeleton of amino acid finally enter the TCA cycle. These all are the significance of the TCA cycle. So, here we stop here. Now, all TCA cycle and glycolysis will be completed. Hello students, today we discuss about the lipid metabolism that comes under the biochemistry. So first of all, what are the lipids? Actually lipids composed of fatty acids, and lipids are insoluble in water. Lipids are insoluble in water. So in this, we mainly discuss about the metabolism. How are they catabolized? How are the lipid anabolized? So this all we cover in this lipid metabolism. So first, lipid. What are the composition of the lipids? Lipids are composed of fats and lipids. Fats means triglycerides or triacylglycerol. These fats store and supply energy to the body. And lipids, phospholipid, glycolipids, cholesterol ester, and this is important to membrane components. This is the two importance for the membrane components. So lipid is insoluble in polar solvents. Polar solvents means like water. Water are a polar solvents and have good solubility in non-polar solvents. Non-polar solvents like benzene, ethanol. This is the non-polar solvents, and these lipids are also a non-polar, and they soluble in non-polar. Non-polar things are soluble in non-polar solvents, and polar are soluble in polar. So lipid is a non-polar, they soluble in non-polar solvent. Lipids are hydrocarbon chain and they having a ester group. Lipids classified as simple and complex lipids. Lipids are classified as simple and complex lipids. Lipids composed of ester of fatty acids. What? They composed of ester of fatty acids. Lipids are composed of fatty acids, and this fatty acids provide energy to our body. Lipid metabolism. In the main function of lipid is they store energy. What are the main function? They store the energy. They structure components of cell membrane and as important signaling molecules. They help in transfer of the signals from one cell to another. So the main function of lipids is they have stored energy, structure components of cell membranes as important signaling molecules. Oxidation of lipids are produced more energy than the oxidation of carbohydrates. What? The oxidation of lipids produce more energy as oxidation of carbohydrates because lipids contain a fatty acids. Human have a fat tissue under the skin, in the abdominal cavity and in the mammary glands. They having a stored fatty tissue. When they burn, when these fats are burned, they provide energy to the cells. Clear? So, first we study about the lipid digestion, then catabolism, and this both process comes under lipid metabolism. So, first digestion. When fat are ingested, they bile salts emulsify the dietary fats in the small intestine and forming mixed micelles. Second step, intestine lipases degrade that triglycerols. Fatty acids and other breakdown products are taken up by the intestinal mucosa and converted into triglycerols. Triglycerols are incorporated with cholesterol and APO2 protein and chylomicrons. These are the chylomicrons, and these chylomicrons move through the lymphatic system and blood stream to tissue. They move through the blood stream to the tissue. Then lipoprotein lipase is activated by APO2 in the capillary, converts triglycerol to fatty acid and glycerol. And this fatty acid enter the cells, and after when they enters the cells they oxidize as fuel or re-esterified for storage, and they provide energy as in the form of ATP. This is the ATP by which we get the energy. Clear?
So digestion or emulsification of fats. Fatty acids can be liberated by simple hydrolysis of the esters bond in triglycerides, but the insolubility of the triglycerides present a problem because digestion occurs following dispersion of dietary fats into small particles with sufficiently exposed surface area for rapid attack by digestive enzyme, right? So what happened? The fatty acids, the lipids having three chain of fatty acids. What? They having three chain of fatty acids. During the synthesis of the lipid, all animal and plants have the ability to synthesize TAG, means triacylglycerol. In animal, many cell types and organs have the ability to synthesize the triglycerides. But the liver and intestines are most active. Liver and intestine are the most active. With all cell types, even those of the brain, triglycerides are stored as cytoplasmic lipid droplet. These triglycerides are stored as a lipid droplet. So, glycerol 3-phosphates are converted into lysophosphatidie acid. What? This glycerol 3-phosphates are converted into a lysophosphatidie acid with the help of fatty acyl coenzyme A. With the help of fatty acyl coenzyme A. Then this lysophosphatidie acid is converted into phosphatidic acids, and this phosphatidic acid converted into a diacylglycerol, and then this diacylglycerol converted into triacylglycerol, right? Then this converted into triacylglycerol, right? This triacylglycerol, from starting glycerol 3-phosphate to triacylglycerol, 4 different types of enzyme are used. The enzymes that are used are glycerol 3-phosphate acyl transferase, acylglycerophosphate acyl transferase, phosphatidic acid phosphohydrolase, diacylglycerol acyl transferase. These 4 different types of enzyme are used in the synthesis of triacylglycerol. Clear? In the synthesis of triacylglycerol. So, triacylglycerol are converted into fatty acid and monoacylglycerol with the help of 2 molecules of water. Clear? So, initial product of digestion is stimulate released by the duodenum of the 33 amino acids peptide hormone pancreazozymin cholecystokinin, that is PCCK. The CCK activity induces emptying of the gallbladder to increase concentration of bile salts and other bile constituents in the intestines, including cholesterol and phospholipids. So, PZ activity causes release of pancreatic digestive enzyme, including pancreatic lipases. Short chain fatty acids up to 12 carbon are absorbed directly through the villi of the intestine, right? Directly they are absorbed at the villi of the intestine. Within the intestinal wall, the triglycerides are resynthesized. These triglycerides are resynthesized by the 2 monoacylglycerol pathway as shown in the right. 2 monoglycerides are converted into 1,2-diglyceride, and these 1,2-diglycerides are converted into triglycerides, right? The great majority of the absorbed lipid, including triglycerides, phospholipid, cholesterol esters, and cholesterol, appear in the form of chylomicron that pass to the lymphatic vessels of the abdominal region and later to the synthetic blood. So triglycerides, cholesterol, and cholesterol esters. Cholesterol esters includes in water and cannot be transported in blood or lymph as free molecules. This lipid assemble with phospholipid These lipids assemble with the phospholipid and apolipids to form a spherical particles that is known as lipoprotein. This is a spherical particle that is known as lipoprotein, and hydrophobic coats that is triglyceride and cholesterol esters. So hydrophilic surface cholesterol, phospholipids and apolipoproteins are present. This is the summary of lipoprotein metabolism, that is intestine dietary lipids that are converted into chylomicrons. These chylomicrons are released and they are converted, go into the liver and converted into triglycerol, cholesterol esters and VADL. Then triacyl glycerol. This chylomicron are also converted into triacyl glycerol, right? And this VLDL also converted into a triacyl glycerol, right? High density lipid, low density lipids, they are converted into high density lipids, low density lipids, and they are also converted into cholesterol and cholesterol ester. And this is the peripheral tissue where they utilized as a energy. Lipid catabolism. In lipid catabolism, fat catabolism, beta oxidation of fatty acids, other oxidation of fatty acid, ketone bodies formation and utilization. We study this all types of food. Means fat catabolism, beta oxidation of fatty acids. So first we study about the fat catabolism. Catabolism, first we study about the lipolysis, that is the fat mobilization. So the triglycerol stored in the adipocytes are hydrolyzed by lipases to produce free fatty acids, and glycerol, which are released to the blood. This process is called as mobilization. The fatty acids, those released diffusively from the adipocytes into the blood, where they binds to the serum albumin. This fatty acids binds to the serum albumin. See, this is the adipocytes, right? This is the adipocyte. This fatty acid, these are the fatty acids. They combine with triacylglycerols, right? They binds with the serum albumins. They go via the blood stream, and after that they present in a monocytes muscles, right? They stored in a monocytes muscles where they utilized as a energy. This is in the form of ATP, that is with the help of beta oxidation, that is citric acid cycle, and they produce energy and release a carbon dioxide, right? So hormone induced fatty acid mobilization in adipocytes. Epinephrine, this is the epinephrine, right? This is beta adrenogenic receptor. When epinephrine is present, the G protein will activate the adenylate cyclase. This adenylate cyclase convert ATP into CAMP. This CAMP is the secondary messenger. They act as secondary messenger. They carry the signals and they convert the pKa into a hormone sensitive lipases, and these hormone sensitive lipases they convert the triacylglycerol into a DAG, then MAG, diacylglycerol, then monoacylglycerol, and then glycerol, and release free fatty acids. This is the catabolism, how the fatty acids are released, right? Hormone sensitive lipases, that HSL. Triacylglycerol lipase is the rate limiting enzyme in the triacylglycerol degradation in the adipose tissue. It's also named HSL because it's regulated by some hormones. Effect of hormones on lipolysis, that is lipolytic hormone, that is epinephrine, norepinephrine, ACTH, TSH, glucagon, insulin, and anti-lipolytic hormone, that is insulin. Glycerol metabolism. Glycerol metabolism takes place where glycerol metabolism will be takes place, that is liver, kidney and intestine. Liver, kidney and intestine. So glycerol, what happened in glycerol metabolism? This is the glycerol This glycerol converted into L-glycerol 3-phosphate with the help of glycerol kinase. Then dihydroxyacetone phosphate, then D-glyceraldehyde 3-phosphate. And after that, this D-glyceraldehyde 3-phosphate goes into glycolysis and gluconeogenesis process, right? And in this process, NADH will be released and ATP will be utilized, right? ATP will be utilized in this process. And the enzyme that are help in conversion of this all process into one to another, that is glycerol kinase. They convert glycerol into L-glycerol 3-phosphate, and glycerol 3-phosphate dehydrogenase that convert L-glycerol 3-phosphate into a dihydroxyacetone phosphate. And triose phosphate isomerase, they isomerize this dihydroxyacetone phosphate into D-glyceraldehyde 3-phosphate. Beta oxidation of fatty acids. Fatty acids are one of the main energy material of human and other mammalian. Fatty acid catabolism can be subdivided into 3 stages. Activation of fatty acids. In activation of fatty acids means acetyl coenzyme synthase, which locates in the cytoplasm, catalyze the activation of long chain fatty acid. This is the long chain fatty acid, and they catabolized with the HSC coenzyme A, and they converted into a acyl coenzyme A with the utilization of one molecule of ATP. And the enzyme that are present is the acyl coenzyme A synthase. Acetyl coenzyme A synthase. Key point of fatty acid activation are they are irreversible and consume 2 phosphates. And the site where they occur is the cytosol. And the second step of beta oxidation of fatty acid is the transport of acetyl coenzyme A into the mitochondria. And carrier is the carnitine. Beta oxidation of fatty acids. Transport of acetyl coenzyme A into the mitochondria, that is rate limiting step. Rate limiting step in means rate limiting enzyme. Carnitine acetyl transferase 1. Carnitine acetyl transferase 1. What happened here? This carnitine is converted into the fatty acyl carnitine with the help of this acetyl coenzyme A, carnitine acetyl transferase 1. They convert the carnitine with the fatty acyl carnitine. This is the outer mitochondrial membrane. This is the inner mitochondrial membrane. This is the cytosol. Here carnitine acetyl transferase is present. This carnitine acetyl transferase is converted into carnitine. Then these are transported as a carnitine with the help of transporter, right? And this carnitine are converted into a fatty acid carnitine inside the mitochondrial matrix. And third stage is beta oxidation of fatty acid. That is dehydrogenate, hydration, dehydrogenate, and thiolide cleavage. So in dehydrogenate, what happened? Fatty acyl coenzyme A that are synthesized in previous fatty acyl coenzyme A that are transported into a transenol coenzyme A. And this process, one molecule of FADH2 is released and acetyl coenzyme dehydrogenase are used as an enzyme. Second step, hydration. From name it is clear, in this process one molecule of water are required. And in this step, what happened? Transenol coenzyme A from the previous steps, transenol coenzyme A are converted into a 3L hydroacyl coenzyme A. 3L hydroacyl coenzyme A. And the enzyme that are used are enoid coenzyme A hydratase. Third step is dehydrogenate. In dehydrogenate, what happened? 3L hydroxyl coenzyme A from the previous step that are converted into beta ketoacyl coenzyme A. That are converted into beta ketoacyl coenzyme A with the help of release of one molecule of NADH. Thiolytic cleavage. Thiolytic cleavage. In thiolytic cleavage, what happened? Beta ketoacyl coenzyme A are converted into acetyl coenzyme A and two molecule of shorter fatty acyl coenzyme A. Two molecule of shorter fatty acyl coenzyme A. Beta oxidation. This is the full step of beta oxidation. This is the palmitoyl coenzyme A. Right? This is the palmitoyl coenzyme A. These are converted into a trans-anyl coenzyme A and then L-beta hydroxyacyl coenzyme A. And this L-beta hydroxyacyl coenzyme are converted into a beta ketoacyl coenzyme, then acyl coenzyme that is myristoyl coenzyme A plus acetyl coenzyme. So beta oxidation pathway in cytosol is a C14 carbon are converted into acetyl coenzyme, C12 carbon converted into acetyl coenzyme, C4 converted into acetyl coenzyme. So overall in beta oxidation pathway, the main conversion of the fatty acids into a acetyl coenzyme A. So the summary of beta oxidation of lipid metabolism is the fatty acyl coenzyme A plus FAD plus NAD plus HS coenzyme A is converted into fatty acyl coenzyme A plus FADH2 plus NADH plus H plus acetyl coenzyme A. Other oxidation of fatty acids: oxidation of unsaturated fatty acid, peroximal fatty acids oxidation, oxidation of propionyl coenzyme A. So first is oxidation of unsaturated fatty acid. That's why occur in mitochondria. In this process, there is isomerization process will be occur from cis to trans and epimerase D minus to L plus. So very long fatty acid chain are first converted into a shorter chain via acyl coenzyme oxidase, and that shorter chain fatty acids are further converted into acetyl coenzyme A with the help of beta oxidation pathway. So propionyl coenzyme A converted into a succinyl coenzyme A. Propionyl coenzyme A converted into a succinyl coenzyme A with the help of enzyme that is carboxylase epimerase mutase. Ketone bodies formation and utilization. Ketone bodies are water-soluble fuel normally exported by the liver but overproduced during fasting or in untreated diabetes mellitus, including acetoacetate, beta-hydroxybutyrate, and acetone. Ketone bodies formation and utilization. The formation of ketone bodies location: hepatic mitochondria, material acetyl coenzyme, rate limiting enzyme is HMG coenzyme synthase. This is the process. Acetyl coenzyme A, thiolase, acetoacetyl coenzyme A, acetyl coenzyme A that is converted into a hydroxymethyl glutaryl coenzyme A, then acetoacetate, then acetone. This is the catabolism. Utilization of ketone bodies at extrahepatic tissue. This is the utilization, how the ketone bodies are utilized. D-beta hydroxybutyrate, succinyl coenzyme A, convert acetyl coenzyme A, succinyl coenzyme A transsulfurase converted into acetyl coenzyme A, then they converted into a 2-acetyl coenzyme A, right? After that, acetoacetate are converted into D-beta hydroxybutyrate. This all step are reversible process. Biological significance. Ketone bodies replace glucose as a major source of energy for many tissues, especially the brain, heart, and muscles during time of prolonged starvation. Normal physiological responses to carbohydrate shortage cause the liver to increase the production of ketone bodies from the acetyl coenzyme A generated from fatty acids oxidation. So fatty acid oxidation are required during the starvation period because they release the energy. Right? They release the energy. This is the hepatocytes, acetoacetate, ketone bodies exported as energy for source for heart, skeletal muscles, right? This is the acetyl coenzyme. They converted into acetoacetate, fatty acid, beta oxidation. When beta oxidation will occur, this fatty acid converted into acetyl coenzyme. This acetyl coenzyme A goes into a citric acid cycles and they converted into glucose. Then glucose transfer as fuel for tissue. Or this acetyl coenzyme A converted into a ketone body formation. This ketone body as exported as energy source for heart, skeletal muscles, kidney, and brain. So beta oxidation are most important during the starvation period. So this all about the beta oxidation, beta oxidation or lipid metabolism. Because beta oxidation is the main process in the process of lipid metabolism. So this all about the lipid metabolism that's come under the biochemistry. Hope this all are clear to you. So today we stop here. Thank you so much.